Right, good evening, Year 11 and some Year 10s. We're coming at you live from Horizon Community College to go through the Biology Paper 1 information. It'll be me and Miss Heslop. Mr. Schuller is manning the comments. Please be aware that comments are on, but you will be blocked if you say anything inappropriate. If you have got any questions, you can ask in the comments, or if you go on Twitter and hashtag AskTeamSci, we'll be able to answer your question for you. So what we're going to do, I'm going to look at Spec 1 and Spec 2 with you. So I'll be taking cell biology and then organisation. Then Miss Heslop is going to look at infection and response, and finally bioenergetics. Okay? So the first part of cell biology is obviously knowing what is inside a plant and an animal cell. So if we draw an animal cell first, you should hopefully recognise some of the structures inside. So if we just put a few dots in. So if we label it up, automatically when you see a cell, you should realise that the large darker part in the middle is a nucleus. Around the outside you have got the cell membrane. In an animal cell it is not a cell wall, it's a cell membrane. You've also obviously got the cytoplasm. This is where the chemical reactions take place. And the last two, which if you are doing a higher paper are guaranteed the ones that you will be asked about, you have got these, mitochondria. I've not left myself very much room. And the other one, the little dots, are your ribosomes. I'm not going to go through the functions of every single one because we're very limited on time. However, the two that you probably will be asked about, you need to know. So ribosomes make protein. Mitochondria, if you ask this one, this is a 2-mark definition. So mitochondria release energy. Never say produce, you can't create or produce energy, so it's release energy in aerobic respiration. So they're the functions that you would more than likely be asked about on a higher paper. If we then look at a plant cell, so if we have a look at a plant cell, these are usually a square shape, and you can identify them because they've got a large vacuole, obviously got a nucleus. They've also got chloroplasts. So as well as having the mitochondria, which are the smaller stripy ones, and ribosomes, they have got chloroplasts as well. So if you are asked about a plant cell, the identifying features of a plant cell, it has got a cell wall. It's got a large permanent vacuole, and they also have chloroplasts. And these are the parts of a plant cell that are different. Chloroplasts obviously absorb light energy, and that's what Miss Heslop will be talking to you about a little bit later on. After that, after knowing the parts of a plant and animal cell, you also need to know how cells are specialised for a particular function, so how they become specialised. So if we just section that little bit off, you should be able to tell me that there are different types of specialised cell. We'll draw a few for you here. Hopefully you'll recognise this one straight away, even though I've got really, really horrific drawing. So you have got here a sperm cell. Identifying features of a sperm cell, obviously got a tail. You need to know why, for swimming. Got a mid-piece. And inside the mid-piece there are lots of mitochondria. If you are ever presented with a cell that has got a lot of mitochondria, you should realise that they then will need lots of energy. So sperm have lots of mitochondria to release lots of energy, which they then use for swimming. In here, at the top, you have got an acrosome, which contains enzymes to digest the way through the egg. And as well, I'll write the number in, there's also a nucleus with 23 chromosomes. Cells become specialised at different stages of their development. There's a slight difference between, well, a massive difference between plant and animal cells. Animal cells, I'll write this down for you, animal cells differentiate at an early stage of development. So animal cells differentiate at an early stage of development. It is different in plant cells because plant cells retain the ability to differentiate throughout their life. And I've just realised I've just been banging on about differentiation and I've not told you what the actual definition is. So differentiation is a process where, get a nice colour, process where cells become specialised for a function. Okay. So that's a little bit about cells as well. I'll get a new sheet of paper. You need to know the differences between prokaryotic and eukaryotic cells. Plant and animal cells are eukaryotic. So plant and animal are eukaryotic, which means they have a nucleus. Bacterial cells are prokaryotic, which means they do not have a nucleus. And if I was to draw a picture of a prokaryotic cell, just do it slightly like this. Don't know why I've drawn a rectangle like a plant cell, but we'll go with it. So like a plant cell, it has also got a cell wall. However, instead of having a nucleus, they have a strand of DNA. You can also call that one chromosome. You're also likely to find little circular loops of DNA within them, and these are called plasmids. Okay? As well as having those features, you might often find around the outside, as well as the cell wall, they've got this slimy layer, and it's called a capsule. Capsules help to protect the bacteria against certain antibiotics and also being engulfed by certain cells. Okay, so the main difference between plant and animal cells are, main difference between eukaryotic and prokaryotic cells are the prokaryotic cells do not have a nucleus. All right. One of the other parts that you could be asked about, I'll stay on the same piece of paper, you could be asked about microscopy. If you attended the session tonight about the required practicals, we covered the required practical about microscopes, so I'll not be going over that now. What you need to know though is the I-am triangle, where I is for image, M is for magnification, and A is for actual size. If you needed to rearrange that, so if you were asked for image, you do actual divided by magnification, times magnification. If you were asked for magnification, you do image divided by actual. When you are measuring, you would be measuring the length of an image with a ruler. So the image is measured with a ruler in millimeters. It's important to measure in millimeters because sometimes you will need to convert to micrometers. To convert from millimeters to micrometers, you need to times by 1,000 because there are 1,000 micrometers in 1 millimeter. So for example, 9 millimeters would then be 9,000 micrometers. Okay? Linked to this topic, there are two types of microscopes. You need to know how microscopy is developed over time. So in the olden days, we had very, very simple microscopes with one single lens. We then developed light microscopes and finally electron microscopes. You could be asked to compare the features of light and electron microscopes. So if we're to have a quick look, A light microscope has got a maximum magnification of times 1500. An electron microscope has got a maximum magnification of 2 million. So obviously, electron microscopes are much more powerful. With a light microscope, the maximum resolution is 200 micrometers, and the maximum resolution of an electron microscope is 200 nanometers. Because it is a smaller resolution, that means it can see more detail, so you can distinguish between very, very slight differences. So 200 nanometers is a distance that it can resolve to. Little bit more information. Light microscopes are obviously very cheap, easy to use, so we use them in school. Electron microscopes are very powerful, but unfortunately you cannot use to look at living organisms. So that's a massive disadvantage. I can't speak. So what we're going to look at next is the last part of Spec 1, which is the second to last part, I need to speed up, is to do with mitosis, the cell cycle, and chromosomes. So if we have a look, chromosomes. Obviously, chromosomes are found inside the nucleus. So if we were to look, you would find chromosomes. So your chromosomes usually go around in pairs when they're matched up. And you have got 23 pairs of chromosomes, which equals in a normal human cell, 46 chromosomes in total. You could be asked how a cell divides and the processes involved in the cell cycle. So if I have a look, if I draw a circle, which represents the full cell cycle, before a cell needs to divide, we've got a stage here which occupies most of a cell cycle, where what is happening is the cell is copying DNA and making more ribosomes and mitochondria. So in this stage, it occupies most of the time, and it's where the cell is getting ready to divide. The next section here occupies a much smaller length of time, and this is known as mitosis, which I will cover very shortly. So mitosis I'll cover in a second. The last part that you need to be aware of is this little part here, and that is where the cell actually divides. So don't confuse mitosis with the process of the cell dividing. It's actually the process where the cell prepares to divide and make sure that it has everything in order and it's copied all its nuclei. So if we have a look at the processes, so in mitosis, we'll make it easy. We will have 2 chromosomes. So the first stage, the cell gets ready to divide. So like we said earlier, the cell will copy its chromosomes. So you'll end up with 4 chromosomes. Then, like chromosomes, will pair up along the middle of the cell. In your next stage, they are, shouldn't have drawn that arrow there, never mind. They are pulled to opposite ends of the cell. So if we just draw a little bigger one. There we go. And in the next stage, the cell will start to cleave, that means to split its cytoplasm. And then eventually the cell will totally split in 2, and you will have 2 identical cells with the same number of chromosomes, which in this case was 2. So that is the process of mitosis. Linked to this very quickly, because I'm conscious of time, you need to know what a stem cell is. So we talked earlier about cells being differentiated. I'll draw a new little box here for stem cells, get a different colour. Not that one, don't like that one, red. A stem cell is an undifferentiated cell which can turn into any type of cell. Okay, so stem cells in humans usually differentiate at a very early stage of development. In plants, you have a special tissue called meristem tissue, and this is in the roots and shoots, and it can differentiate into lots of different types of cells. So if you were to take a cutting of a plant, you could actually plant it again, and the tissues would differentiate into root cells, shoot cells, things like that. There's one last part about stem cells, therapeutic cloning, so I'll very quickly do it on the back. If you do separate science, this will make more sense to you because you actually cover cloning. In therapeutic cloning, it can be used to treat diseases such as diabetes, paralysis, thanks Miss Heslop, my mind went blank, and another P1, Parkinson's. So, in therapeutic cloning, what happens, you have got a patient, so this is our poorly patient, and you take a normal body cell from the patient. So cell from patient. You take the nucleus out, and what you do, you get an egg cell from anyone, from a donor. So egg from donor. You keep that nucleus, and what you do with this one, you get rid of the nucleus, and you end up with an empty egg, and you fuse those together. So the nucleus out of the patient is fused into the egg cell from a donor. That means that the genes inside this nucleus are exactly the same as the ones from the patient. You then let that develop into a ball of cells, or you can also call it an embryo. And these cells can then be inserted into the patient to help cure certain diseases. So you take one cell out, put it into the patient, and it would help. Maybe if they had damaged pancreas, you could put it into the pancreas, and it would differentiate into a into cells that actually work, and that is called therapeutic cloning, and that's the process of it. Right, going over time. Last little bit, I'll just give you the definitions. So you need to know about transport in cells. You obviously should know this, so I'll just give you the definitions. So in transport, there are 3 definitions that you need to know. You need to know what diffusion is. You need to know what osmosis is. And you need to know what active transport is. So we'll take diffusion first. You should hopefully all be saying this out loud because you should know what it is. Diffusion is the movement of particles from an area of high concentration to an area of low concentration. That's the definition for that. And very quickly, there are factors that can affect it. So the factors that can affect diffusion: surface area, temperature, distance, and if we're talking about an animal, things like blood supply. The better the blood supply, the quicker diffusion happens. That's diffusion in a nutshell. Obviously, there is more that you need to know about that one. I have just not got. Thomas Johnson, what is a nucleus? I've just seen that. What an absolutely ridiculous question. Come on, we know this. I've lost my train of thought because I got so angry then. Right, osmosis. So, really, right, more? Love a bit of dilute pop. Such sensible questions from set 1. Right, osmosis is the movement, you can say diffusion, of water from a dilute to a more concentrated solution through a partially permeable membrane. Obviously, there's a required practical to do with that and to do with potatoes. Last definition in transport, active transport, is the movement of particles against their concentration gradient using energy. So diffusion and osmosis do not require energy. They go with the concentration gradient. Active transport does require energy. It moves against the concentration gradient. Two examples of it are glucose in the small intestine and mineral ions from the soil. So there are your two examples, right? I'm really going to have to speed it up. Miss Heslop's staring at me thinking it's her turn. So organisation then, has that focused in or something gone wrong here? Yeah. Oh, Mr. Schuller, it looks like it's not focusing properly. Is it because I've got too many sheets on? Let's see. There, much better, thank you. So in organisation, very quickly, your body or a plant are organised into cells, then tissues, then organs, then organ systems, then a whole organism. For example, pick an organ first. I could say the heart. The heart might be made of muscle tissue. What's muscle tissue made up of? It's made of muscle cells. The organ system that the heart is in is the circulatory system. And the whole organism might be a human, okay? The main system that you're going to be asked about is the digestive system. I've not really got time to go over the full digestive system. I just want to pick out a few key points about organisation. So the main points really in this topic, you have got to know what an enzyme is and you must know the functions of the main enzymes. So an enzyme is a biological catalyst. So biological catalyst, they are made of protein and they speed up reactions. They can be denatured by two things. So they can be denatured by temperature and pH. Denature means that the active site changes shape. And what I mean is, when you have an enzyme, it'll have a very specific shape that will allow a substrate to go into it, to bind into it. If you denature an enzyme, it loses the shape of its active site so that the substrate can't bind. Substrate cannot bind. And like I said, there are three important enzymes that you need to know. So just section this off a little bit here. So the main ones, amylase. Amylase breaks down starch to glucose. Protease breaks down protein to amino acids. And lipase breaks down fats into 3 fatty acids plus glycerol. These are all examples of metabolic reactions. They're all reactions that happen within your body. Miss Heslop's going to talk to you a little bit about that later. Okay? Last little bit on digestion before we move on. You also need to know what bile does. Bile is made in the liver, stored in the gallbladder, and it's got two functions. Number one, it emulsifies fat, and it also neutralizes acid. I'm going over this very briefly, so this is the bare minimum that you need to know just because I am conscious of time. Okay, what we're going to do, there is also, in organisation, you need to know about the lungs and gas exchange. We haven't really got time now to go over the lungs and gas exchange. I'm going to skip over this, but you need to know the main structure of the lungs and also the process of ventilation in your lungs. So I'll leave that. If we get time, we will come back to it at the end. What I want to very quickly talk to you about is the heart. This is new to the biology specification or to the trilogy science specification. So you need to know the main chambers of the heart and also the main blood vessels. As you look at the heart, it is a mirror image. So this is the left side, this is the right. And you need to know what the parts of the heart are and the blood vessels. So this one is the left atrium. This one is the left ventricle. Obviously, it's a mirror image. The opposite side is the right atrium. And this one is the right ventricle. They're the main chambers of the heart. You also need to know the blood vessels leading to and from the heart. So blood enters the heart through the vena cava. It then enters the right atrium, right ventricle, and it leaves up and out of the heart through the pulmonary artery. I'll use a different colour. I've used blue because it is deoxygenated blood. It then enters the heart from the lungs, where it picks up oxygen through the pulmonary vein, enters the left atrium, into the left ventricle, then up and out of the heart through the aorta. They're the different parts of the heart, and that's the pathway of blood going through the heart. As well as this, you need to know the features of the main blood vessels in your body. So if we have a look, these are pictures of the main blood vessels. The easiest one to identify is the capillary. Capillaries have a wall that is a single cell thick. This is to allow diffusion between the blood and body cells to take place. The other easier one to identify is the artery. Arteries have thick, muscular, elastic walls in a small lumen or middle because they carry high pressure blood. And finally, the other one then is veins. A vein has a thinner wall, a larger lumen, and they have valves. So when the blood leaves the heart, it goes through an artery because arteries go away from the heart. Think of the A. Goes through the arteries to the body cells in capillaries, then the blood enters the heart or goes towards the heart through a vein. Last little bit on heart, blood and, dropping pencil over the floor. Blood vessels, you need to know the main parts of blood very quickly, briefly. You have got red blood cells. Obviously their role is to carry oxygen. You have got, here's a different colour, you've got white blood cells that can just look like a cell with a large nucleus. White blood cells, they produce antibodies. And the other two parts, here's a different colour, I'll draw them up here. The little fragments of cells are called platelets. And these clot blood. And then the other part, the final part that you need to know about, just drawing a yellow squiggle, is the plasma. So the plasma is the liquid part of the blood, and it transports things like antibodies and CO2 around the body. Okay, last little bit on the organisation. We're going to have a look at plant organisation. There is a little bit more in organisation about health and things like that, but to make sure that we've got time, I will skip over that. Is that okay with you, Miss Heslop? So last little bit on organisation that you need to know about, because we're conscious of time. You need to know about how plant cells are organised. So if we have a look, so this is a cross-section of a leaf. The top layer, you have got the cuticle. The cuticle is waxy, which helps to reduce water loss. Here you've got the upper epidermis. This is thin and transparent, which allows light to pass through. The next layer here, you have got the palisade layer, and this one has got lots of chloroplasts, which absorb light. The next layer down, you have got the, written it wrong, thank you, Miss Heslop, I just couldn't think of a word. You've got the spongy mesophyll. This has got air spaces, which allow diffusion. So that part there is an air space. And then at the bottom layer is called the lower epidermis. And in the lower epidermis, you have got guard cells. And the job of these cells is to open and close this one, which is the stomata. The stomata allows gases into and out of the leaf, which will allow photosynthesis to take place. As well, through the stomata, you also lose water, and that is to do with a process called transpiration, which we'll look at very briefly. Okay, last little bit here, you've got a vascular bundle, and in the vascular bundle, you've got your transport tissues, which are xylem and phloem. That links into transpiration and translocation, which I will very, very briefly cover. So let me just get rid of some of these. So transpiration this is the evaporation of water vapor from the leaves through the stomata. In transpiration, the water moves through the plant in the xylem. Translocation then. Translocation is the movement of dissolved sugars from the leaves to other parts of the plant. If you are ever asked on a diagram to label the xylem and phloem, it will label one part of it for you, and you could be asked to label the other part. So if I just write quickly here, this is just an example of what it might look like. If it labels this one and says that that is the xylem, you will automatically know that the other part must be the phloem. So last little bit, xylem is water and minerals. Phloem is sugars. You can also say amino acids, but it's easier to remember sugars. So the rest of Spec 2 is about CHD and health issues, risk factors, things like that. As well, there's a little bit on cancer, so I'll just very quickly talk you through it. I'll not be writing anything down, but you just have a listen. In coronary heart disease, fatty material builds up in the arteries that lead towards the heart. This restricts blood flow to the heart, which obviously carries glucose and oxygen. If the heart muscle is not getting glucose and oxygen, it can go into heart attack because it cannot respire. In coronary heart disease, there are lots of different treatments. The main two that you might be asked about are statins and stents. Statins are drugs that lower cholesterol, which means that less fatty material might be built up in the arteries. However, you have to take them every day for the rest of your life, and there can be severe side effects, especially with the liver. The other treatment is stents. Stents are put into blocked arteries and the little metal cages which hold them open. Obviously, the main advantage of this is that the blood can pass through to the heart. Disadvantage-wise, it is a small operation. There's obviously an infection risk, and what else am I going to say? It can lead to blood clots because sometimes it can tear the tissue. Other little part about it, I think the main bit to mention about cancer, you need to mention when you're talking about cancer, it is due to uncontrolled cell division. If you are asked how a tumour forms, it is uncontrolled cell division. There are two types of tumour, benign and malignant. Malignant are the ones that spread, and they are spread by cells breaking off, and these travel in the blood and they can form secondary tumours. Okay? Right. I've gone slightly over, and there is a lot more content in the first two specs, so you will need to be looking at your revision guide. It's just a bare minimum what we've been going over today. So we're going to have just a 2-minute break just whilst Miss Heslop sets up to look at the next part of the paper. Okay, so if you've got any questions, don't forget to hashtag AskTeamSci.
Right, okay, year 11, back with spec 3 then. So infection and response. First of all, I'm just going to mention the difference between a communicable and a non-communicable disease. So a communicable, oh God, spelling, means that the disease can spread, and non-communicable obviously means that it doesn't spread. Okay. Next then, a definition for a pathogen. So a pathogen is a microorganism that can cause disease. And you have got 4 types of pathogens. Okay, so the 4 pathogens, you've got bacteria and viruses, which we'll be talking in more detail about. And then you've got protists and fungi. So bacteria, viruses, protists, and fungi. And then you need to know a bit more about how bacteria and viruses cause you harm, and then you learn about different diseases to do with each type of pathogen. So I'll go over how bacteria and viruses can cause you harm. So both of them reproduce rapidly make sure you use the word rapidly, inside your body. And then bacteria release toxins. And viruses live inside your cells and damage your cells. Okay, so don't forget they both do the one in green, but they both do something separate. Next then, you learn about different diseases for each type. Oh, what's so I'm going to start with bacterial diseases and then we'll look at the rest. So bacterial disease, these are nice and easy. You've got salmonella, which is food poisoning. You get this from undercooked meat, or if you do not wash your hands and you touch raw meat and then you touch cooked meat. I'm going to put causes. Or you could talk about contaminating your meat that you've cooked with uncooked meat. And then prevention is really simple. Wash your hands or wipe down your work surfaces with disinfectant, or you can just say cook food thoroughly. Okay, and your second bacterial disease is gonorrhea. Miss, can you help with the spelling? Gonorrhea?
Gonorrhea. Do you want me to spell it out?
Yes, please.
Right. G-O-N-O-R-R-H-O-E-A.
Lovely, thank you. Okay, and obviously this is caught through sexual contact, so unprotected sex. Prevention, obviously wear a condom. And if you do get a bacterial disease, I'm going to talk about this in more detail later, but obviously then the main way of treatment for bacterial diseases is antibiotics, but I'm going to come on to talk about antibiotics later. Moving on then. Oh, thank you. So, viral diseases. There are 3 that you have learnt about, 2 in animals and 1 in plants. So we have got HIV. I know I can remember that one. I can't remember tobacco mosaic virus.
I think maybe only 2.
No, they've got another one.
Measles.
That's it. Thank you. So
we've got a few comments to this. Apparently we need to slow down a little bit.
Oh, sorry, it's me.
Guys.
I'm always too fast and they always tell me to slow down. Do you want me to put these back under?
I think we'll just take
I'll go slower.
Take a bit slower.
Right, so HIV, again, can be caught through unprotected sex or sharing of needles. My class always shouts at me for going too fast. And the prevention would obviously be to, again, have protected sex, don't share needles. And the big thing about HIV is that it affects your immune system. And therefore, if you get another infection, you are less likely to be able to fight it off. Measles then causes a rash all over your skin. It's caught through if somebody sneezes or coughs on you. And therefore, if you have measles, you are just suggested to stay away from other people. And then TMV, I'm going to do this in a different colour because this is just for plants. So it stands for tobacco mosaic virus, and therefore when you will know if your plant has this because it has a mosaic pattern on the leaves. Okay, and this is spread through contamination of gardening tools. And therefore you are just told to basically, obviously, don't share your gardening tools between an infected plant, or wash your gardening tools, well, I should say disinfect, in between. And if you have a viral disease, the way you treat it is with antiretrovirals. And again, I'm going to talk about treatment in more detail later. Just turn this over. So next thing, I'm going to talk about protist disease. You only learn about one of those, which is malaria. Okay, and please remember, it's not a mosquito that causes malaria. The mosquito is the vector, which transmits it between people. Now, I'm going to try to draw you. I'm just going to draw a little fly instead of a mosquito. Okay, so remember, the mosquito is the vector. It will then come along and bite a human. And then when it goes into the human, when the protist is transported into the human's blood, the first place that it goes is the liver. And then secondly, it will go into the red blood cells to reproduce. Okay, now you have to know about how it reproduces differently in a human and in the mosquito. So when it's in the human, it reproduces asexually. But when it's in the mosquito, it reproduces sexually. Okay, and another thing just to note is that it's always a female mosquito. Some prevention methods then, so obviously to try stop from getting malaria, you would use firstly a mosquito net. You could then spray insecticides which will kill the mosquitoes. Or you could talk about draining pools of stagnant water. So that the mosquitoes haven't got anywhere to reproduce. And then lastly, we're going to go on to fungal diseases. And again, you only need to know one of these, which is a plant one, which is rose black spot. So as the name suggests, if your plant has rose black spot, it will get black spots on it. And this one is spread through water. Okay, and basically the treatment for this one is if your rose bush has rose black spot and you see a leaf that's contaminated, you just need to pull off infected leaves. Right then, moving on. We're going to look at the human defence systems. So firstly, we'll talk about the non-specific human defence system. Okay, so your body prevents you from getting pathogens entering your body by 4 different things. So 1, simply just your skin, which is a protective layer. 2 is hairs. specifically in your nose, which trap the pathogens. Next is your stomach acid, which will then kill, hopefully, any bacteria that get inside. And last is cilia, which again trap, I'm going to put cilia in trachea, which trap the pathogens again. Okay, so that's your non-specific human defence system. That's about preventing them entering your body, but obviously sometimes they do get inside your body, and that is when your immune system which is your white blood cells, are then going to come along and remove the pathogen. So you've got two types of white blood cells. You've got lymphocytes and phagocytes. Okay, so before I just talk about the white blood cells, I'll just remind you about what an antigen is. So every cell has antigens on their surface. And the definition of an antigen is a unique marker. So if a cell that's foreign gets into your body, then your white blood cells will recognise that the antigens are foreign and that it needs to get rid of that cell. Okay, so this is when, firstly, your lymphocytes will start to produce antibodies. And the antibodies are the complementary shape to the antigen so that they can bind. These will then stick. So they bind to the antigen and then they stick the pathogens together so that then they will all be in one place. And then that is when your phagocytes come along. So then the job of the phagocytes, and you must use this word, engulf the pathogens. Okay, so moving on to vaccination. So remember, vaccination is about prevention.
Sorry, Miss, we've asked to go. Could you go back to the last one? There's a couple of us asking back to the last bit and just talk to you about the last couple of bits on there.
This bit?
Yeah, please.
Right, so antigens are unique markers on the outside of every cell. You've then got two types of white blood cell, which are going to come along and hopefully get rid of these pathogens. So lymphocytes are the ones that work first. They produce the antibodies, which are the complementary shape to the antigens. So they bind to the antigens and cause all the pathogens to stick together. So obviously, if they're all in one place, that means it's going to be easier to get rid of. And then once they've all stuck together, the phagocytes then come along and engulf the pathogen. And it's really important that you use the word engulf, which basically means they take them inside of them. Okay, but remember this word is important. Okay, so vaccination is the prevention of a disease, and this is where you inject a dead or inactive pathogen. Okay, that will then stimulate your white blood cells. I'm going to put WBC for short, white blood cells, to produce antibodies. And then if you are reinfected, so if you then get that pathogen in the future, your body has memory cells which remember the pathogen and they can produce antibodies really quickly or rapidly. You must use the word rapidly. So it can produce antibodies rapidly. Okay, and you must use that word. Right, next then, I said I was going to come back to talking about antibiotics. So, antibiotics, remember, only treat bacteria. And you have to know why they cannot treat viruses. So the reason they cannot treat viruses... Am I still going too fast?
Just still a little bit.
I usually just tell them to shut up and make sure I'm speeding. I'll try, I'm sorry. So they only treat bacteria. I'll just keep repeating myself. And the reason they can't treat viruses is because viruses live inside cells. I think it's because I write fast, so I feel like I'm just going at my speed.
I can play it back, miss.
Yeah, true. They live inside cells, so they are inaccessible, which means they can't get to them. I can't spell inaccessible.
Two C's.
Able or able?
Able.
Inaccessible to the antibiotic. So basically that just means that they can only treat bacteria because the viruses are inside the cells, which means they cannot get to the virus. If they got to the virus, it would mean damaging your cells at the same time, which you do not want. Okay, I've already said that if you're treating a virus, you use antiretrovirals. And lastly, just a little bit about painkillers. Let's do it over here. Remember, painkillers, it says it in the name, they only relieve the pain. They don't actually treat you. So they don't get rid of, spelled that wrong as well. They don't actually get rid of the pathogen, they are just helping with the pain. Give you all a minute.
Catch up time.
Okay, so I've already written that side. Moving on then to look at discovery and development of drugs. So remember, most drugs originally come from plants and microorganisms. Okay, but now obviously we can chemically synthesize drugs in a pharmaceutical lab. Now don't forget about, oh I've just realised the time and I've not gone to spec 4 yet, but never mind.
Sorry, just keep going.
So in a pharmaceutical lab, you have to do obviously drug tests before. We have to test for 3 reasons. So first reason is to test for safety or side effects. Do not use that as 2 different answers. They are the same answer. Okay. Second one is to check if it actually works, which is called efficacy. So if it works. And finally, we have to test for the dosage. See what the correct dose is. So you have to go through different stages of drug testing. So firstly, you have pre-clinical trials. That means before humans. And it must be tested on animals or tissues. And firstly, that is to see if it is safe. Then it will move on to clinical trials, which means humans, testing it on humans, of healthy people, and a small number of them. And again, this is to see if it's safe on humans and for side effects. And then finally, if it's safe on that number of small healthy people, you can move on to a large number of patients with the disease. And this is when you do a double-blind trial using a placebo. So first of all, the definition of placebo is it contains no active drug. You must say that if you are talking about a placebo. And then the double-blind trial means that neither the doctor or the patients know who is getting the actual drug and who is getting the placebo. Run out of room. Right then, and then we're going to be moving on to spec 4, bioenergetics, which is photosynthesis and respiration. Yeah. Right then, so photosynthesis to start with. We'll start with the basics, so the equation. So you obviously need water plus carbon dioxide, which gives you glucose and oxygen. Okay, and the symbols. So remember, glucose is C6H12O6. And to balance the equation, you don't have to physically work it out. You just have to remember to put a 6 in front of the water, the carbon dioxide, and the oxygen. Okay. You then need to know what the plant uses glucose for. So you'll learn, because obviously the whole purpose of photosynthesis is to produce glucose for the plant. First one is respiration. Next, making proteins. Making cellulose, which is in the cell walls. And then you can store it as different things. So store it as starch or store it as fats and oils. Now, Miss Nerissa obviously mentioned this earlier to do with chloroplasts. So please remember, when you're talking about photosynthesis, in order for it to work, you also need light energy. And you must say the word light, you can't just say sun. So light energy, which is absorbed by chlorophyll inside the chloroplasts in the leaf. Okay, you then need to know about things that can speed up the rate of photosynthesis. So firstly, having more light, and you must use the word more carbon dioxide, more chlorophyll, and then a higher temperature. Okay, and this brings us on to limiting factors and some limiting factor graphs that you need to know about. So limiting factors. Okay, you've got our light intensity, carbon dioxide concentration, and temperature. Okay, now the light intensity and the CO2 concentration graph are exactly the same. So you'll have light intensity at the bottom and rate of photosynthesis up the side. And then what happens is they increase and then they level off. And that is because at the start, the light is the limiting factor. But at the end, it is something else. And that something else would either be CO2 or temperature. So something else is the limiting factor. Okay, and remember the CO2 graph is exactly the same, apart from you would put CO2 at the bottom, same rate up the side, exactly the same with exactly the same answers, but where you put light here, you would change your answer to carbon dioxide. The temperature is slightly different. So it comes to a peak and then decreases. So temperature and rate again up the side. And then you would have an optimum temperature. And then this time it increases up to the optimum and then decreases. And you would have to say because at too low a temperature, there is not enough energy. But at too high a temperature, enzymes denature, which Miss Neresi talked about earlier. Okay, moving on then, we're going to go on to respiration. So aerobic respiration first. Again, equation first, it's just the opposite from photosynthesis. So this time you're using oxygen plus glucose, and that gives you water plus carbon dioxide. Okay, and the symbols are all exactly the same with the exact same numbers in front. So C6H12O6, 6H2O, and 6CO2. Okay, aerobic respiration then. Big thing, it uses oxygen. So when we come on to look at anaerobic respiration, that's the big difference. This one uses oxygen, anaerobic does not. Okay, so some key things. It happens in the mitochondria, which again, Miss Neresi spoke about earlier. It releases more energy than aerobic, so it's more efficient, anaerobic, sorry, more efficient than anaerobic. And again, uses oxygen when anaerobic does not. So if you are then asked about what happens during exercise, basically everything just increases. You are going to need more energy. So remember, the whole purpose of respiration is to release energy. So exercise, you're going to need more energy. Therefore, you're going to need to do more respiration. Always use the word more. Then you're going to need more oxygen and glucose. And you're also going to produce more of the products. So water and carbon dioxide. Okay. So that your muscles can contract. Someone's having a right similar. So anaerobic respiration. We're nearly done, don't worry. This happens in the cytoplasm and produces less energy, so it's less efficient. Okay, and this time in animals, remember there is no oxygen. Main difference. This time the equation is just glucose turns into lactic acid. And remember, lactic acid is poisonous, which causes cramp. Okay, now that's in animals. In plants and yeast, anaerobic respiration is slightly different. Okay, you just have glucose, but this time the products are different. Turns into carbon dioxide plus ethanol. So that is why we can use yeast, which is also another name for anaerobic respiration is fermentation. And we can use the carbon dioxide produced by this to make bread, or the ethanol to make alcohol. Now I'm just going to go back to it in animals. This is higher tier only, so if you are sitting a foundation paper, you don't actually need to know this. But when you do anaerobic respiration, you produce an oxygen debt, and that is why you keep breathing heavily after exercise, because you have to repay the oxygen, and then to break down the lactic acid, it travels to the liver, where the oxygen will then break it down back into its products. Okay, but remember that is higher tier only. Last thing is metabolism. This is going to be very important, so please make sure you're paying attention. So metabolism definition equals the sum of all the chemical reactions in your body or in a cell. Okay, so metabolism means the sum of all the chemical reactions. It is not just about digesting food. You then need to know examples of metabolic reactions. So any example of any reaction happening in your cell or in your body is an example of metabolic reaction. So you could literally say any reaction that you know that happens. So just give you a couple of examples. So key one, respiration. If you're talking about plants, you could talk about photosynthesis. Okay, you could also talk about the formation of lipids from 3 fatty acids and a glycerol. You could talk about conversion of glucose to starch. Okay, but again, any chemical reaction that happens in your body is an example of a metabolic reaction, but I've just given you 3 there that are really important. Right, year 11, that is the end of the revision. If you are set 1, you need to stay on because I'm going to go through the set 1 only stuff with you, but for everybody else, you can now go and have a break. So 2-minute break, but set 1, stay on. Right, okay, so set 1 only stuff. Just move these out of the way. First of all, I'm going to be, obviously this won't necessarily flow in a correct order because it's all just from either spec 1 or spec 2 randomly. So, culturing microorganisms, this is about aseptic technique. Okay, so I'll talk you through the process. So, you are trying to produce a pure, uncontaminated culture of bacteria. So, first of all, you need to be able to explain why you are doing each step. So, you would get an agar plate, which has been sterilised before use. Okay, and that is to obviously make sure that there are no bacteria on it. You then need to sterilise your inoculating loop, again to make sure that there's no bacteria going to contaminate your culture. Then you're going to dip the loop into your bacteria. And spread it on your agar plate. And when you are doing this, you need to make sure that you do not take the lid off before you need to. So only remove the lid for as short a period of time as possible. And again, this is all about making sure that no other bacteria can get onto your culture. Okay, and then you will, if you are just growing the bacteria, then you would stop there and you would tape the lid shut. But you also did the practical where you talk about antiseptics and testing different ones. I talked about that in the required practical session after school, so I'm not going to go over that in as much detail. So I'm just going to jump to tape the lid shut. And remember, you don't tape it all the way around, you just tape it a little bit each side because you need oxygen to get in so that anaerobic microorganisms cannot grow. So tape the lid shut, and again, that's just to make sure that no bacteria get in or out. It's all about making sure that your culture is a pure culture. Okay, so I'll quickly talk about the results. So imagine you get an agar plate. What you are looking for is, if you have tested an antibiotic or an antiseptic, you should see a clear zone around that, and that is when the bacteria have been killed. Make sure you say they've been killed, and they have grown, but then they've been killed after. And you are looking for the largest area around that. So remember, area, we're bringing it into maths, pi r squared. So you have to measure the diameter that way, measure the diameter that way, and then work out an average diameter. And then from that, take your radius and do pi r squared to work out the area of the clear zone. The one with the largest area could obviously show you that that is the best antibiotic or antiseptic, but then remember, you had a question in your mock that said, why might that not be the case? And you have to make sure you've tested sort of the same concentrations. You need to know if it's safe to use in a home, things like that. Okay, so that's that one, and I did go over that quite a lot in the required practical session. Next then, monoclonal antibodies. So first of all, I'll talk you through the process of producing monoclonal antibodies, and then we will talk about the uses of them. So firstly, when you are making a monoclonal antibody, you need to inject a mouse with a specific antigen. Okay, the mouse, it's lymphocytes, and hopefully you should know what I'm talking about because I've just been over this. The lymphocytes from the mouse will start to produce antibodies against the antigen. Okay, you then take the lymphocytes and you fuse them with a tumour. And this is going to form something called a hybridoma cell. You are then going to clone the hybridoma, so you'll end up with lots of them. So imagine you've got one hybridoma cell. You're then going to clone, hence the name monoclonal antibodies. So basically you'll end up with lots of these hybridoma cells, okay, and then you will take out the antibodies and culture them. Remember, that just means grow them. And then you can use them for different things, which I'll talk about now. So different uses. So firstly, we've talked quite a bit about them being used in pregnancy tests. And then they can also be used in the diagnosis and treatment of diseases. Okay, so the reason they can be used really well, so I'll talk about one quick advantage and disadvantage. So the reason they're good at diagnosis and treatment of diseases is you can specifically target them. So an advantage is you can specifically target the cells you want to. So for instance, if you're trying to target cancer cells, you can make them directly go to the cancer cells because they will be targeted rather than killing any of your own cells. But a disadvantage is that they have foreign proteins in from the mouse and therefore they might be rejected. And then last thing is plant diseases. So first of all, you have to know how to identify a plant disease. So if you're growing some plants, you want to look out for things like leaf discoloration. I'm really bad at spelling tonight. Discoloration? E-R or A-R?
O-R, I think.
O-R. Oh, right. Discoloration. Stunted growth. I think you should have said that louder, Mr. Shaw. Any patterns on the leaves. So things like, obviously we talked about rose black spot, if you've got black spots or mosaic patterns on your leaves. And the way you can identify which disease you have got is just to look in a gardening manual. Or you could send it off to a lab and ask them to do some tests. Okay, so next thing I'm going to talk about nutrient deficiencies. So there's two that you learnt about. We've got nitrate deficiency and magnesium deficiency. So magnesium deficiency produces yellow leaves. Nitrate produces stunted growth and yellow leaves. And the reason being for nitrates is that nitrates are used to make proteins. So if you are deficient in nitrates, then you are not being able to make enough proteins. And magnesium deficiency, magnesium is used to make chlorophyll. Have I?
No,
Oh, me.
someone's mocking your spelling when Chelsea Brown can't spell either.
No, she's spelling it the way Miss Heslop
All
has.
right, I'll pipe down.
Why, which word?
Discoloration, very interesting spelling.
Well, never mind. You don't get marked on spelling, kids, in science. So magnesium makes chlorophyll, and therefore if the chlorophyll is not made properly, you're not going to be able to, the leaves will turn yellow. Last thing about plant diseases is you need to know about how plants defend themselves. So plant defences. And plants can defend themselves in three ways. So you've got physical defences, mechanical defences, and chemical. Thank you. Okay, and an example of each one of these. So a physical defence would be something like hairs or spines or stingers on the outside. Okay, to deter from being eaten. Mechanical would be things like a waxy cuticle, or the really strong cellulose cell walls. And chemical is about the fact that obviously some plants can be poisonous. I'm going to have to spell poisonous now. Poisonous, which obviously if they're poisonous, they are not going to be wanting to eat them. And I believe that is everything. Can you think of anything else?
No, I think that's about it. Well done, Miss Heslop. Did really well. Right. So that's covered everything that you should need in very brief detail for your biology paper 1. Obviously, we haven't had time to go over every single little bit of information. We will be taking pictures of the slides that we've made and we'll post them onto our Twitter and Facebook Horizon channels. So the next session that we're going to do will be Wednesday night from 6 o'clock, and that will be chemistry with Mr. Addy, Mr. Schuller and I. Okay, thank you very much for tuning in. Mr. Schuller, have you got anything to say before we go?
No, that's it. Thank you very much.
Thank you, you've been a delightful audience.
You seen that?